The discovery of DNA began with the identification of the cell as the basic unit of life in the 1830s by Matthias Schleiden and Theodor Schwann. However, it was not until the late 19th and early 20th century that scientists began to understand the chemical composition of cells. In 1869, Swiss biochemist Friedrich Miescher discovered a substance in the nuclei of white blood cells, which he named nuclein. Later, it was determined that nuclein was actually DNA, but at the time, scientists did not fully understand its significance. In the 1920s, British scientist Frederick Griffith conducted experiments with two strains of bacteria, one that caused pneumonia in mice, virulent, and another that did not, non-virulent. He discovered that when heat-killed virulent bacteria were mixed with living non-virulent bacteria, the non-virulent bacteria became virulent. This phenomenon was later termed transformation. In 1944, Oswald Avery, Colin MacLeod, and Macklin McCarty identified that the transforming agent in Griffith's experiment was DNA. This was a groundbreaking discovery that demonstrated that DNA, not proteins, was responsible for carrying genetic information. The double helix structure of DNA was first proposed by James Watson and Francis Crick in 1953, building upon the work of Rosalind Franklin and Maurice Wilkins, who used X-ray crystallography to obtain images of the structure of DNA. Watson, Crick, and Wilkins were awarded the Nobel Prize in Physiology or Medicine in 1962 for their discovery of the structure of DNA. Since then, the study of DNA has revolutionized many fields, including genetics, medicine, forensics, and evolutionary biology. The Human Genome Project, completed in 2003, provided a complete map of the human genome, further advancing our understanding of DNA and its role in life processes. DNA, short for deoxyribonucleic acid, is a molecule that carries the genetic instructions for the development, function, growth, and reproduction of all known living organisms and many viruses. It is a long chain-like structure made up of smaller building blocks called nucleotides, which are connected to each other through covalent bonds. The nucleotides are composed of a sugar molecule, a phosphate group, and a nitrogen-containing base, which can be one of four types: adenine, A, guanine, G, cytosine, C, or thymine, T. The sequence of these bases along the DNA molecule encodes the genetic information, similar to the way letters in a book encode information. The specific sequence of DNA bases determines the characteristics of an organism, such as its physical traits, susceptibility to certain diseases, and other biological functions. DNA replication is the process of copying the genetic information stored in DNA, which is essential for cell division and the transmission of genetic information from one generation to the next. If you think this video is informative, please give thumbs up.